Welcome to the SQANTI3 tutorial video from the CONISA YouTube channel. SQANTI3 is the leading software for long-read defined transcriptome quality control and filtering. In this video, we'll explore the key features of SQANTI3. The first part is an overview of SQANTI3, briefly introducing the functional isotranscriptomics pipeline. We'll also give you a summary of how SQANTI3 works and introduce the recommended long-read processing workflow before SQANTI3. The second part of the SQANTI3 tutorial details how to run the quality control step. We will show you all the input files for running SQANTI3 and look at how SQANTI uses those files to classify isoforms, perform quality control, and generate output files. After quality control, we will show you how to perform filtering with SQANTI3. Users can filter their transcriptome based on their desired criteria using rules or machine learning filters. Also, we will walk you through all the output files in the filtering steps. Finally, we will demonstrate how to use the SQANTI3 Rescue feature, which aims to recover potentially valid transcripts discarded as artifacts due to a lack of confident validation during the quality control step. Now, let's start. To better understand SQANTI3, we need a basic idea of the functional isotranscriptomics pipeline. SQANTI3 constitutes the first module of the functional isotranscriptomics pipeline, which is an end-to-end strategy to perform isoform-level bioinformatics analyses. The SQANTI3 tool is designed to enable quality control and filtering of long-read defined transcriptomes, which are often rich in artifacts and false positive isoforms. Therefore, a good curation of the transcriptome is indispensable to proceed with functional isotranscriptomics analysis and produce valid, biologically sound conclusions or hypotheses. After generating a high-quality transcriptome with SQANTI3, downstream steps include using ISO-ANNOT to annotate isoform models, including positionally defined functional features such as motifs, domains, etc. Please note that ISO-ANNOT, a tool for de novo annotation of isoforms, is currently under development. However, users can run ISO-ANNOT Lite within or outside SQANTI3 to impute functional features from other already annotated transcriptomes. Expression-based functional analysis using TAPIS. TAPIS is a Java graphical user interface application that leverages expression and domain or motif annotation information to gain insight into the functional implications of alternative isoform expression. After learning the basic idea of the functional isotranscriptomics pipeline, the next question is how SQANTI3 works. SQANTI3 is a tool for the in-depth characterization of isoforms obtained by full-length transcript sequencing, commonly returned in a FASTA or GTF file format. SQANTI3 combines the long redefined transcripts with the reference annotation and other orthogonal data to provide a wide range of descriptors of transcript quality. SQANTI3 is mainly designed to perform two different tasks, both of them equally important. The first is SQANTI QC, including isoform classification and quality control for long-read. Utilizing the SQANTI3 categories and subcategories and a long list of transcript-level attributes and descriptors, users can carefully inspect the properties of their isoform models and identify potential problems generated during library preparation and raw data processing. The second is the SQANTI Filter for artifact removal. Using the large number of descriptors calculated by SQANTI3 QC, users can make informed decisions to remove potential false positive isoforms from their transcriptomes. This is particularly relevant considering the biases and pitfalls of current long-read sequencing protocols. Recently, however, we have implemented a final step in the SQANTI3 workflow. Reference Transcript Rescue, SQANTI3 Rescue, to find matching reference transcripts for discarded artifacts. This module intends to keep the diversity in the transcriptome, for example, preventing loss of transcripts with, mostly, valid junction chains for which a long-read defined isoform could not be validated. By running the rescue, SQANTI3 will select a set of reference transcripts to which the discarded artifacts can be confidently assigned and add them to expand the filtered transcriptome. Here is our recommended workflow, including the best way to generate the SQANTI3 inputs and how to proceed after SQANTI3 quality control and filtering. Let's start with the library processing based on PacBio single-molecule real-time sequencing. Based on your budget, we have different recommended workflows.
Here is an example of the standard cost workflow.
The workflow starts by collecting samples from several experiments. After that, both long-read and short-read sequencing is conducted for each sample from the experiment. We recommend building a single transcriptome per experiment using the long-reads data, and the short-reads data is used to quantify the transcriptome. Finally, you can get a transcript expression matrix with quantification of each sample. In this workflow, you can quantify long-read defined transcriptome using short and long-reads data. If you have a lower budget, we recommend another workflow. For long reads, we recommend pooling all samples for each experiment and only sequencing for the pooled samples. In other words, pooling a sample means mixing several samples or replicates in an experiment together in a batch. For short reads, we recommend sequencing for all samples. The transcriptome model is built based on the long reads pooled sample data. Finally, you can get a transcript expression matrix with quantification of each sample. However, in this workflow, you can only quantify long-read defined transcriptome using short-reads data. After getting raw long-read sequencing data, the next step is to process the data. We recommend using your preferred transcriptome building tool, like the IsoSeq method. The IsoSeq method is an end-to-end command line tool for sequencing and analyzing full-length transcript isoforms shown in this animation. We do not recommend using SQANTI3 on raw long-reads data, as it is not designed as a tool for raw long-reads data quality control. The collapse of isoform models is important in building a transcriptome model. Typically, long-read data processing pipelines generate a large number of highly redundant isoform models. We recommend collapsing these using tools such as cDNA Cupcake or Tama Collapse to reduce the number of isoforms and create unique isoform models before running SQANTI3. In quality control and filtering, we strongly encourage users to inspect their long-read defined transcriptomes as carefully as possible, including filtering their transcriptome to remove potential false positive isoforms, which are abundant in long-read generated transcriptomes. Quantification of the filtered transcriptome using short or long reads and your preferred tool. We do not recommend using the expression estimates input into SQANTI3 for downstream analysis. These are used for quality control purposes only. Once all artifacts are removed from the transcriptome, the reads can be used to obtain more accurate quantification. As introduced before, the first step in the SQANTI3 is the SQANTI quality control. To make it runs, you need to prepare several files. Let's introduce how SQANTI3 quality control works from the input files. The minimal file requirements are as follows. The first one is a long-read defined transcriptome file. It can be obtained after processing data from third-generation sequencing technologies like IsoSeq from PacBio or Oxford Nanopore. SQANTI3 is compatible with any long-read-based transcriptome building pipeline output, such as IsoSeq3, Talon, or Flare. SQANTI3 accepts it in several formats, such as FASTA, FASTQ, and GTF. By default, SQANTI3 expects the transcriptome to be provided as a GTF file, and we recommend sticking to this format if your transcriptome construction pipeline allows it. Using a GTF file can skip the mapping step to improve the speed. If you provide your transcript sequences in FASTA or FASTQ format, you must also supply the FASTA option. In this case, an additional mapping step will be initially performed. SQANTI3 currently supports Minimap2 by default, Ultra, Desalt, and GMAP for mapping. To select a mapper, supply the name to the aligner_choice argument. The second one is a reference transcriptome annotation in GTF format. This file will be used as a reference to describe the novelty across long-read transcripts. Finally, a reference genome is needed in FASTA format, for example, HG38. Before running SQANTI3, make sure of the following. The transcriptome annotation GTF must match the provided reference genome. The chromosome or scaffold names must be the same in the reference annotation and the reference genome. You can also provide optional input data to help SQANTI3 quality control generate comprehensive results, which can finally help to filter false positive isoforms and contribute to downstream analysis. The first file is the short reads data file. SQANTI3 quality control uses short read data to validate several aspects of long read defined transcripts. For example, to compute gene or isoform expression, junction coverage, and TSS ratio. Short read data can be supplied to SQANTI3 in several ways. The first one is raw short read data for users who may want SQANTI3 to compute all short read based attributes internally without the need to perform any additional preprocessing. Short read FASTA or FASTQ files can be provided using the short_reads argument. Alternatively, users may supply preprocessed short read data in one or more of these formats. Short read-based transcript expression is one of them. Users can supply their pre-computed isoform expression via the expression argument. Depending on whether this information is provided as one or multiple files, two formats are supported. Suppose you input one expression file per sample. In that case, you can provide several expression data files as a chain of comma-separated paths or provide a directory where only expression data is present. Accepted formats include the output of Callisto and RSEM. Alternatively, it is possible to provide a pre-computed expression matrix. The matrix should be a tab-separated file with transcripts as rows and samples or replicates as columns. Transcript identifiers must match the ones included in the input long read-defined transcriptome, and the name for that column must be ID. The rest of the columns in the header can be named as desired and should correspond to the sample or replicate identifier. The next is splice junction coverage by short reads. If you have short read data, you can run star to get the junction file, usually called sj.out.tab, and supply it to SQANTI3 via the C argument. It is possible to use a different mapper. However, SQANTI3 requires that the junction format be similar to star's output format. The final one is short read BAMs, which should be obtained by mapping short reads against the reference genome using a splice-aware mapper, such as star. SQANTI3 QC will use this information to compute the TSS ratio. The second file is the cage peak data file. The peaks are used as anchors to define promoters and as units of promoter-level expression analysis. This is particularly relevant given that 5' RNA degradation can generate variability in the transcription start site that can be mistaken for a novel transcription start site. Cage peak data must be provided as a bed file, where column 1 contains the chromosome. Column 2 contains the 0-based index start coordinate. Column 3 contains 1-based index end coordinate. Column 6 contains strand information. The bed file needs to be supplied to SQANTI quality control via the cage_peak argument. The third one is the poly-A motif list file. We have included a list of mouse and human data motifs in the data/poly-A_motifs/ folder. Please let us know the most likely poly-A motifs for other organisms or clades through the comments below. The full-length count information file is next, which supports full-length counts from the PacBio isoseq pipeline provided through the FL_count argument. There are mainly two acceptable formats. The first one is the single-sample full-length count. A single-sample full-length count file is automatically produced by the isoseq with mapping pipeline with the following format. The next one is the multi-sample chained full-length count file, which is produced by the Cupcake and will have the following format. The final one is the TAPAS annotation file. TAPAS has been created specifically to provide downstream analysis, visualization, filtering, and ad hoc query tools for working with RNA-seq data at the gene and isoform levels. When the ISO-anodlite flag is activated, a GFF3 file containing isoform-level functional annotations for a reference transcriptome can also be supplied via the GFF3 flag, which helps you transfer isoform-level functional feature annotations to the SQANTI3 quality control output. By doing this, functional feature information will be added to your long-read-defined transcriptome, unlocking the TAPIS functional and feature-level analyses.
After getting the SQANTI minimal inputs and some additional inputs,
you can start to run SQANTI3 quality control. Now, I will introduce how SQANTI uses those input files to do quality control. As explained before, the first part in SQANTI3 is SQANTI QC, including isoform classification and quality control for long-read-defined transcriptomes.
SQANTI3 classifies each isoform by finding the best matching reference transcript in the following order.
The first one is the full splice match, meaning the reference and query isoform have the same number of exons, and each internal junction agrees.
The exact 5' start and 3' end can differ by any amount.
The full splice match has several subcategories shown as follows. Reference match is transcripts with less than 50 base pairs distance to annotated transcription start sites and transcription termination sites. Alternative 3' end is transcripts with more than 50 base pairs distance to annotated transcription termination sites. Alternative 5' end is transcripts with more than 50 base pairs distance to annotated transcription start sites. Alternative 3' and alternative 5' end is transcripts with more than 50 base pairs distance to annotated transcription start sites and transcription termination sites. The second category is the incomplete splice match, meaning the query isoform has fewer exons than the reference, leading to a partial match of the splice junctions, but each internal junction agrees. The exact 5' start sites and 3' end sites can differ by any amount. Incomplete splice match also has several subcategories, and let us have a look at it. The first one is the 5' fragment, which means transcripts have fewer 5' exons, and the reference agrees with all its junctions. The second one is the 3' fragment, which means transcripts have fewer 3' exons, and the reference agrees with all its junctions. Then, the internal fragment means that transcripts with all junctions agree but have fewer both 5' and 3' exons. The next is the intron retention under the incomplete splice match category, means that transcripts have at least one exon covering at least two adjacent reference exons.
Finally, monoexon under the incomplete splice match category means that transcripts only match one reference exon with a multi-exon reference.
Then, let's look at two novel isoform categories, novel in catalog and novel not in catalog. They are subcategories based on whether they use a combination of known junctions, a combination of known splice sites, or at least one splice site is novel. There are several subcategories in novel in catalog.
For example, there are two different reference isoforms: reference isoform 1 and reference isoform 2.
The combination of known splice junction subcategories combines the same splice junctions from isoform 1
and isoform 2.
Then the second subcategory in the novel in catalog is the combination of known splice sites. Transcripts under this subcategory have novel combinations of existing splice sites from the reference. If a transcript uses a combination of known junctions or a combination of known splice sites and includes intron retention, they are categorized in the novel in catalog intron retention. Finally, a novel in catalog monoexon subcategory contains transcripts with one reference exon and a combination of known splice sites. The next novel category is the novel not in catalog, in which the query isoform does not have a full or incomplete splice match and has at least one splice site that is not annotated.
The next is genic intron, in which the query isoform is completely contained within an annotated intron.
Genic genomic is the category for the query isoform that overlaps with introns and exons. Instead of classifier isoforms in different SQANTI3 categories, SQANTI3 has several quality control descriptors using the additional complementary data.
Let's first start with the quality control of the transcription start site.
SQANTI3 checks how far a detected transcription start site falls from the reference transcription start site. Then, SQANTI3 uses cap analysis gene expression cage peaks data to distinguish between trustworthy and untrustworthy transcription start sites. This is particularly relevant given that 5' RNA degradation can generate variability in the transcription start site that can be mistaken for a novel transcription start site. The method is used for studying gene regulation through quantitative analysis of transcription start sites. By sequencing the 5' ends of cDNA-converted capped RNAs, the cage peak enables the identification of core promoter regions and 5' ends transcriptional activity. Therefore, the cage peaks are used as anchors to define promoters and as units of promoter-level expression analysis. SQANTI3 quality control will use this data to validate the transcript's transcription start sites. First, it will evaluate whether the transcription start site of the different long-read-defined transcripts falls inside a cage peak. Then, it will calculate the distance to the nearest detected cage peak. All those information is used to distinguish between trustworthy and untrustworthy transcription start sites. Now, look at how SQANTI3 quality control for transcription termination sites. SQANTI3 QC first checks how far a detected transcription termination site falls from the reference transcription termination site. Then, if users provide the poly-A motif or employ A site data, SQANTI3 QC will use this data to create several descriptors regarding the presence of the transcription termination site within poly-A motifs or peaks and the distance to them if detected. A poly-A tail is a 10 to 30 nucleotides downstream sequence that consists of a 6-nucleotide signaling site called the poly-A motif. In human cells, the most common one is A, T, A. If a poly-A motif's data file is provided, SQANTI3 checks if a poly-A motif is inside the file at the 3' ends of the transcript. In addition, SQANTI3 returns the sequence and the distance to the closest poly-A motif detected. Complementary to poly-A motif information, poly-A site data can be supplied. The poly-A site data is typically generated through sequencing mRNA 3' ends. More than a dozen protocols capture this information, and for humans, mice, and worms, you can download public poly-A site data from the poly-A site atlas. Using this information, SQANTI3 will determine the distance to the closest poly-A site and whether the transcript's transcription termination site was found inside a poly-A site. Then, I will introduce the quality control for the splice junction. SQANTI3 quality control first checks whether the splice junction is known or novel. Then, SQANTI3 contains whether the splice site is canonical or non-canonical. The canonical splice sites are those described initially and most commonly found in up to 99% of introns and have GT at the donor site, just after the 5' ends of the cut, and AG at the acceptor site, just before the 3' ends of the cut. If you have short-read data, you can run star to get the junction file and supply it to SQANTI3 introduced in the SQANTI3 quality control input part. Short-read coverage across junctions represents an informative measure for quality control of sequenced transcripts, and SQANTI3 finds whether short reads support the junction. SQANTI3 quality control descriptors also check intrapriming. Now, let's look at what is intrapriming and how SQANTI checks it. Oligo-DT primers are designed to prime the poly-A sequence at the 3' ends of the mRNAs. However, besides priming at the 3' ends of mRNA, the oligo-DT primer can also prime the poly-A sequences present internally within mRNA. The initiation of cDNA synthesis from the oligo-DT primed at both locations results in the generation of 2 truncated cDNAs from a single mRNA template. This process is called intrapriming. SQANTI3 detected it by checking internal A-rich regions. Finally, SQANTI3 quality control finds the switching of the reverse transcriptase event called RT switching. This event could happen as follows. When reverse transcription starts, an RNA with 2 direct repeats is a potential template for switching the reverse transcriptase from one repeat to the other. This may be enhanced by a secondary structure bringing into the vicinity of the repeats. The resulting cDNA molecule possesses only one copy of the direct repeats, which appears as novel junctions. SQANTI3 uses the following RT switching algorithm to decide an RT switching artifact. The algorithm locates 8 nucleotides direct repeats, which is characteristic of reverse transcriptase switching between the upstream mRNA boundary of the non-canonical intron and the intron region adjacent to the downstream exon boundary, with a slide of 1 nucleotide upstream or downstream. Now, let's introduce the SQANTI3 output files. As a result of running SQANTI3 QC, the tool will create a series of output files in the specified director. Given that SQANTI3 also runs several other tools internally, a series of subfolders will also appear in the output directory. The SQANTI3 output has 2 main categories. The first one is the main output. Now, let's first look at the classification file in the main output. A classification file is a tab-separated file where transcripts or rows and quality control attributes computed by SQANTI3 are columns. Isoforms are identified by their ID in the input long-read GTF file. The classification file contains 48 columns, including all quality control attributes of a transcript. Here is a detailed explanation for some hard-to-understand columns. The junctions file is another SQANTI3 output file, a tab-separated file containing each junction information for each isoform. Rows are identified by the isoform ID and the junction number. For example, the first junction in the isoform PB.83093.1 is named junction_1. The second is called junction_2, and so on. Here is a detailed explanation for some hard-to-understand columns. Now, let's look at the other category of SQANTI3 quality control output, transcriptome-related files. GTF file, the _corrected.gtf file constitutes the main transcriptome output of SQANTI3. It contains the annotation for all transcripts supplied to SQANTI3. This file may be used for downstream analyses such as quantification. However, we recommend filtering your transcriptome using the quality control attributes in SQANTI3 before proceeding with your analyses. For this purpose, you may apply one of the 2 filters available in SQANTI3 or, alternatively, define your own filtering criteria. FASTA file, the _corrected.fasta file contains the genomic sequences of transcripts supplied to SQANTI3 QC, obtained using the _corrected.gtf file and the provided reference genome. FAA file, the _corrected.faa file contains FASTA formatted protein sequences derived from transcript sequences. These sequences are obtained by running the GeneMark Open Reading Frame Prediction algorithm and will not be output if the SkipORF flag is supplied. CDSGFF file. The _corrected.gtf.cds.gff file is generated by expanding the GTF file to include coding sequence information obtained from the GeneMark Open Reading Frame predictions. This means that, in addition to exons and transcripts, coding sequence will also be included as a feature in the last GFF column. TAPIS GFF3 file. If the isoanodlite argument is used, isoanodlite will be run internally, and a TAPAS formatted GFF3 file containing all isoforms in the input transcriptome will be generated in the output folder. This allows direct usage of a SQANTI3 process transcriptome in TAPIS. Of note, if a pre-annotated TAPIS GFF3 file is supplied to SQANTI3 QC via the GFF3 flag, the TAPIS GFF3 annotation will also contain functional features transferred by position. The SQANTI QC report is generated by the SQANTI3_report.r script, which takes the classification and junctions files from the SQANTI QC main output. The report includes over 150 plots, therefore, to ease interpretation, we recently implemented the report argument with 4 options: PDF, HTML, both, and skip. This means that the report can be created as an interactive HTML document. Or a PDF report. Or missed entirely if the user does not wish to see a report for the quality control run. Users proficient in R are welcome to modify the R script to add new figures, and we will constantly be adding new figures. SQANTI QC also generates some supplementary output files. Find the details of those files from following URL. After the SQANTI quality control, artifacts remain in the resulting transcriptome. SQANTI removes artifactual transcripts by applying a rules classifier or machine learning classifier based on the attributes obtained through SQANTI3 quality control. Now, let's first look at how to running the rules classifier. SQANTI rules will be used to accept or discard an isoform based on the attributes obtained through SQANTI3 quality control. To define those rules, the user will need to create a JSON file specifying which SQANTI3 quality control attributes and thresholds are to be used for filtering and, therefore, the properties of the transcripts that are to be included in the final transcriptome. The rules filter only needs an SQANTI3 classification file to work. To run it, execute SQANTI3_filter.py, providing the rules argument followed by the path to the SQANTI3_classification.txt file, like the following. The user needs to create filter rules, and if no rules are provided, a default set of rules for the filter will be used. Now, I will introduce how to make your own filter rules. Specifically, for each SQANTI3 structural category, like a full splice match, incomplete splice match, or novel in the catalog, the user should build an array of rules. A rule is made up of one or more requisites that must all be met for an entry to be considered a true isoform, evaluated as an AND operation. If multiple rules are created for the same category, they are treated as separate and independent, evaluated as an OR operation, so the transcript must pass at least one of these rules. If rules are not set for a specific category, they can be defined using the REST category name. In this example, we have a custom filter defined in a JSON file. The filter specifies criteria that isoforms must meet in order to be kept. For isoforms in the full splice match category, the filter requires that they not be a potential intra-priming product, as determined by having less than 60% in the percent of genomic adenines in the downstream 20 BP window. For the incomplete splice match category, the isoform must be larger than 2 kilobases and shorter than 15 kilobases and must be cataloged within the 3'_fragment, 5'_fragment or internal_fragment subcategories. The novel in the catalog category requires all splice junctions to be canonical or covered by at least 10 short reads. The novel not in the catalog category requires all splice junctions to be canonical and the absolute distance to the closest annotated transcriptional start sites and transcriptional termination sites must be 50 BP or less. Or covered by at least 10 short reads, and the absolute distance to the closest annotated transcriptional start sites and transcriptional termination sites must be 50 BP or less. For the rest of the categories, the transcripts will be kept if they meet the following conditions: not suspected of being an RT switching artifact, and all of their splice junctions are considered to be canonical. The transcript is coding, it is not an intrapriming product, and it is not a monoexon transcript. Instead of SQANTI3 rules filter, SQANTI3 incorporates an automated filter based on machine learning. A random forest classifier is designed to discriminate potential artifacts from true isoforms without needing user-defined rules or manually set thresholds. Similarly to the rules filter, the machine learning filter can be run using the SQANTI3_filter.py script by providing the ML argument followed by the path to the SQANTI*_classification.txt file as follows. The classifier is trained with true positive and true negative sets, either provided by the user or the built-in options. By default, true positive is reference match, and true negative is novel non-in-catalog with non-canonical junctions. The true sets will be split for training and testing the random forest model, by default, 80% for training and 20% for testing. After getting the labeled training and testing data, the random forest classifier is built through the following steps. Step 1. Building decision trees. The first step in creating a random forest classifier is to split the training data into smaller subsets. A decision tree is constructed by finding the best feature splits that separate the isoforms into their correct class labels using the Gini impurity. Step 2. Combining results. The next step is to use each decision tree to make a prediction for an isoform. Then, all the predictions are combined to form the final result, using methods like majority vote. Step 3. Evaluating accuracy. The final step is to evaluate the accuracy of the model. This is done by testing the model on a separate set of test isoforms and comparing the predictions to the actual labels. This is the workflow of the random forest classifier in SQANTI3 machine learning filter. Let's now look at the overview of the SQANTI3 machine learning filtering. From the plot, we can find instead of the random forest classifier, the SQANTI3 machine learning filter script also includes a threshold-based filter to eliminate intrapriming artifacts. Users can specify the maximum percentage of adenines at the transcription termination site using the intrapriming or I argument. Transcripts with a percentage of adenine nucleotides in the genomic sequence immediately following the end of the transcript greater than the specified value will be flagged as intrapriming artifacts. By default, transcripts with more than 60% adenines at the 3' ends of the genome will be flagged. A transcript will be labeled as an isoform if it passes both the machine learning filter and the intrapriming filter. Finally, let me introduce the SQANTI3 output files. The first one is a classification file. The result_classification.txt file is a new classification file that contains an extra column labeled filter_result. This column classifies all the entries as either isoform or artifact. The second one is an inclusion list. The inclusionlist.txt file is a text file that lists the IDs of the isoforms that have passed the filter. Lastly is a filter report. The SQANTI3_filter_report.pdf is a PDF report that includes various plots to describe the performance of the filtering process. Other specific output files are based on the filter method chosen by the user, either rules or machine learning, and can be found on our GitHub wiki. SQANTI3 Rescue is a new module added in SQANTI3 version 5.1 for keeping the diversity in the transcriptome. The algorithm is designed to recover transcripts in the reference transcriptome using evidence from discarded isoforms in the SQANTI3 filter steps. The goal is to avoid losing transcripts that are detected as expressed by long-read sequencing but not validated using orthogonal data, which leads to the loss of transcriptome diversity. SQANTI3 Rescue will assign each discarded artifact to the best matching reference transcript. As a result, The expanded transcriptome GTF will include a set of reference transcripts and the long read-defined isoforms that pass the filter. Now, let's discuss the steps involved in the SQANTI3 rescue algorithm. Step 1A is automatic rescue. The automatic rescue strategy in SQANTI3 is designed to recover full splice match isoforms. To achieve this, the first step is to retrieve all reference transcripts that were represented by at least one full splice match in the original post-quality control transcriptome. This information is available in the associated _transcript column of the classification.txt file. If all full splice matches with the same associated _transcript are removed, it's assumed that the transcription start sites and/or transcription termination sites of the long read-defined transcript is different from that of the matching reference transcript, which leads to the loss of the diversity of that reference transcript in the transcriptome. In this case, the associated _transcript from the reference is rescued, and the discarded full splice match is not rescued. This is the automatic rescue process. Next, I will introduce how SQANTI rescues other non-full splice match supported references. Step 1B selection of rescue candidate and target transcripts is the first step of rescuing other non-full splice match supported references. In this step, the rescue strategy considers other isoforms including incomplete splice match, novel in the catalog, or novel not in the catalog artifacts. For incomplete splice match artifact transcripts, there are two possible situations. When the same discarded reference transcript is supported by both the full splice match and incomplete splice match, the incomplete splice match artifact is collapsed into the rescued reference transcript during the automatic rescue step. When the reference is only supported by one or more incomplete splice matches, these incomplete splice match artifacts that constituted evidence of a non-full splice match supported reference will be included in the rescue candidate list. For novel transcripts from the novel in the catalog or novel not in the catalog artifacts categories, since there is no associated transcript information, all transcripts classified as artifacts will be included in the rescue candidate list. Finally, all reference or long read-defined transcripts from genes that have at least one rescue candidate are considered rescue targets. In the second step of the SQANTI3 rescue workflow, we aim to match each rescue target with its corresponding rescue candidates. To do this, we use a tool called Minimap2. In Minimap2, we consider the rescue targets as the reference genome and the rescue candidates as reads. The following command line is used to run Minimap2. The mapping hits, which are pairs of candidates and targets obtained during the mapping process, are extracted from the output SAM file. This includes both primary and secondary alignments. Now, let's look at the third step of the SQANTI3 rescue process. In this step, we will be validating our reference rescue targets before considering them for inclusion in the transcriptome. To validate our reference targets, we first need to run SQANTI3 quality control on the reference transcriptome and supply the output classification.txt file to SQANTI3 rescue using the K-flag. It's essential to use the same orthogonal data files as when running SQANTI3 quality control for the long read transcriptome, as the rescue strategy is based on the assumption that the same evidence is required to validate all rescue targets. Once we have supplied the reference classification file, SQANTI3 rescue will apply the SQANTI3 filter to the reference transcriptome. The filter that will be applied will be specified by the rules or machine flags used when running the rescue. So, if we run SQANTI3 machine learning based filter, we should also run the rescue using the ML option, and if we run the SQANTI3 rules filter, we should also use the rules option. That's it for step 3 of SQANTI3 rescue. Let's move on to the next step. In the final rescue step, which we'll call rescue by mapping, we combine the mapping hits, the candidate target pairs obtained during mapping, and the results of the SQANTI3 filter applied to the reference transcriptome to generate a final list of reference transcripts to be rescued. This step is divided into 3 tasks. The first one is the filtering of targets. We start by removing any candidate target pairs where the rescue target failed the filter, either the machine learning or rules filter. If you're using the rules filter, this means that the target transcript didn't pass the rules defined in the JSON file. If you're using the machine learning filter, this means that the target transcript didn't reach the specified machine learning probability threshold. However, if you're using the machine learning filter, only the target transcripts with the highest machine learning probability will be selected for rescue. The second one is selecting reference targets. During mapping, both long-read defined transcripts and reference target transcripts are considered. As a result, rescue candidates can match either of these transcripts. From this point forward, only targets selected from the reference transcriptome will be considered to avoid adding redundant transcripts to the final transcriptome. For example, if a rescue candidate's best match is another long-read defined transcript from the same gene, there's no need to add another transcript as a rescued representative, since the best matching transcript is already included in the transcriptome. The third one is removing redundant reference transcripts. During the rescue, it's possible that some of the reference transcripts we want to include in our transcriptome are already represented by a full splice match from the same gene or have already been retrieved during the automatic rescue. In this case, reference target transcripts that are already represented by a long-read defined isoform should not be included in the transcriptome to avoid adding redundant transcripts. Essentially, this means that the artifact's best matching transcript is already represented by an isoform in the transcriptome. After applying this final filter to the list of rescue targets, SQANTI3 Rescue outputs a list of rescued reference transcripts, which are then added to the long-read transcriptome GTF. And that's it. SQANTI3 is a powerful tool for characterizing and filtering long-read defined transcriptomes, with its main focus on isoform classification, quality control, and artifact filtering. The recently added Reference Transcript Rescue module offers an additional step to ensure that no valid transcripts are lost in the filtering process and to expand the filtered transcriptome. With its wide range of descriptors, comprehensive reports, and ability to combine data sources, SQANTI3 is a valuable tool for anyone working with long-read transcriptome data. Thank you for watching this tutorial, and we hope you found it helpful.